I'm going to be talking to you today about the contribution of epithelial to mesenchymal transition and embryonic process to various aspects of tumor heterogeneity and disease progression. And what we have learned about this concept using single-cell analysis of circulating tumor cells enriched from metastatic breast cancer patients. As primary tumors grow, they become vascularized, and this together with increased tumor cell motility and other complex processes results in the intravasation of tumor cells into the circulation. A majority of these tumor cells, the CTCs, are destroyed through apoptosis, and the viable ones extravasate at a suitable distal site where the competent ones form metastases which are ultimately lethal to the patient. The tumor cells that lodge at these distal sites continue to evolve through the acquisition of additional mutations and through changes in their gene expression landscape. But monitoring these changes is quite complicated due to the inaccessibility of these sites and the risks associated with repeat biopsies. These sites, however, are vascularized and the tumor cells continue to shed molecular information back into the blood. This can be in the form of DNA fragments, proteins. RNA is not stable in the blood, therefore RNA fragments, as well as lipids and metabolites. Membrane vesicles such as exosomes harbor certain components of this molecular information, but CTCs basically harbor the entire complement of molecular information that is present within tumor cells. So therefore, isolation and characterization of the circulating tumor cells can provide a non-invasive tool to repeatedly and rapidly monitor tumor evolution over time. However, CTCs are extremely rare. They are estimated to be present at a frequency of 1 tumor cell within 1 billion blood cells, and therefore their isolation poses quite a technological challenge. Given their clinical utility, many different approaches have been developed to enrich CTCs from the blood of cancer patients. These can rely on the biophysical properties of these cells, such as size and density, or the biological properties which define the proteins that are expressed on their surface. This forms the premise for antibody-based CTC isolation, which can be used in the positive selection mode, where antibodies are directed against proteins that are expressed exclusively on the surface of the tumor cells, but not on the white blood cells. Or it can be used in the negative depletion mode, where antibodies are directed against white blood cells, thereby deflecting them away from the CTCs that can be enriched. The current CTC isolation device that we use in the lab is dependent on this negative depletion approach, and we collect the patient blood and incubate it with antibodies directed against the white blood cells. These are conjugated to immunomagnetic beads, and the mixture is processed through a microfluidic CTC chip, which has 3 functional compartments. In the first segment, the red blood cells, the platelets, and the unconjugated beads are removed from the blood using a size-based sorting, the hydrodynamic cell sorting, leaving behind the nucleated cells, which is a mixture of the CTCs and the tagged white blood cells. This then enters the second segment of the chip, which is a series of curved channels where the fluid flow centers them in single file. In the third segment, magnetic force is applied, thereby deflecting the white blood cells away from the CTCs, which are collected in the product chamber. This is the CTC isolation instrument where this microfluidic CTC chip is loaded. Processing blood through this chip basically removes the red blood cells and the platelets away, and the white blood cells away from the CTCs, which are then collected in the product chamber. This process is very gentle, so the CTCs are intact and viable, and they can be used for various downstream applications. They can be stained and visualized by high-resolution imaging. They can also be cultured ex vivo. The efficiency is relatively low, but these cultures are extremely unique because they come from patients with late-stage disease, and therefore they harbor unique mutations that are found only in patients who have been treated extensively with various therapeutic regimens, and they can be genotyped and subject to drug sensitivity assays, and they can also be used for in vivo studies to understand mechanisms of metastasis. The cells are intact, and therefore the RNA is of very high quality, and we can use tumor-specific RNA-based molecular signatures to look at the tumor load in the blood. We can also pick these cells and subject them to single-cell analysis, such as single-cell RNA sequencing, single-cell proteomics, as well as single-cell metal-seq to define the inter and the intra-tumor heterogeneity within each patient, as well as across a cohort of cancer patients. Tumor heterogeneity comes in many flavors. There can be differences in cell lineage, where epithelial tumor cells coexist with mesenchymal tumor cells. There can be differences in proliferation, where proliferative cells coexist with quiescent cells. We can also have differences in growth factor signaling and kinase activities in these tumor cells. In fact, in our CTC cultures established from metastatic breast cancer patients having received multiple courses of therapies, we found that there were HER2 positive cells coexisting with HER2 negative tumor cell populations. This was not due to HER2 amplification, and these populations had very different properties. They had different tumorigenic properties and different drug sensitivity profiles. And then, of course, there is the genomic heterogeneity that arises from differences in mutational burden, as well as amplification and so on. These are not isolated states. There's quite a bit of interaction between these cell states. In fact, the HER2 population is proliferative, whereas the HER2 negative population is quiescent. Epithelial to mesenchymal transition is incompatible with simultaneous proliferation, and when these two processes occur at the same time, there is extensive mitotic defects which lead to genomic instability and genomic heterogeneity. These epithelial to mesenchymal transition serves as a great model to understand many steps that occur during tumor metastasis. The evidence supporting a role for EMT in metastasis comes from cell culture and mouse models, and clinical evidence supporting a role for EMT in metastasis was quite limited due to the limitations of antibodies used to detect these two states in in tumors. It is an embryonic process. It occurs during wound healing, cancer invasion, and also has been implicated in resistance to various types of therapies used to treat cancer patients. So using a highly sensitive RNA in situ hybridization method, which allowed us to use multiple probes against epithelial and mesenchymal states, we applied this to CTCs isolated from metastatic breast cancer patients, and we observed that EMT does not represent two distinct states, but instead is a continuous process with multiple intermediates. Before I get into this slide, let me actually color code it for you. The red and the orange represent CTCs, which are more epithelial, and the blue and the purple represent CTCs that are more mesenchymal. And each bar represents CTCs that were enriched from a patient. So the first thing that you can notice is that there's extensive heterogeneity in the lineage of these CTCs circulating in the blood of breast cancer patients. In addition to this heterogeneity, the CTCs also switch from one state to another during treatment. Let me illustrate that using this index patient from whom we had multiple blood draws. When the patient first came to us, the patient was progressing on previous therapies, had high numbers of CTCs, and most of them were mesenchymal. The patient was treated with a combination of inhibitors against PI3 kinase and MEK. The patient responded, the number of CTCs declined, and they also switched their lineage to an epithelial phenotype. When the patient progressed, the numbers increased, and you can see there's increasing CTCs that are in the mesenchymal state. And this cycle actually continues, where the cells continue to oscillate between epithelial and mesenchymal states depending on where they are in drug response. We wanted to see whether we could actually detect EMT in CTCs using single-cell RNA sequencing data that we had derived from over 106 CTCs, over 100 CTCs derived from a cohort of breast cancer patients. When we applied an EMT signature to this single-cell RNA sequencing data, we found that half the CTCs were epithelial and the other half was mesenchymal. This allowed us to actually study the molecular properties of these two populations of CTCs. We found that the epithelial CTCs were more proliferative compared to the mesenchymal CTCs. In addition, they also expressed increased levels of growth factors such as the estrogen receptor, HER2 expression, HER2, and the androgen receptor. So as I mentioned, the epithelial CTCs have increased levels of estrogen receptor expression and HER2 expression. And they also expressed increased levels of AR expression and have increased AR signaling, which may actually contribute the increased proliferative potential of these epithelial CTCs. Unsupervised clustering of multiple single CTCs derived from metastatic breast cancer patients showed that there was extensive heterogeneity within this population, with some classes of CTCs expressing high levels of the hypoxia genes, actin cytoskeletal genes, and platelet-derived genes. And about 1/3 of these CTCs expressed high levels of mRNAs that code for structural ribosomal proteins. Applying GSC analysis to this subset of CTCs, we found that they had increased translational processes happening within these CTCs. We also observed that the CTCs with increased expression of mRNAs coding for structural ribosomal proteins were more epithelial compared to their counterpart. The CTCs also had increased proliferative signatures, that is, the ones that expressed increased levels of mRNAs coding for these structural ribosomal proteins. They also had increased levels of V2F targets, which are required for DNA replication, cell cycle progression, and DNA damage responses in cells. Now, mTOR activation plays a major role in driving mRNA translation, and we wanted to see whether these mTOR activity would be higher in epithelial CTCs, which also exhibit increased levels of these mRNAs coding for structural ribosomal proteins. So we looked at two different single-cell RNA sequencing data set from CTCs derived from breast cancer patients. We first of all divided these CTCs or classified them as epithelial and mesenchymal, and we found that in fact the mTOR activity was much higher in the epithelial CTCs compared to the mesenchymal CTCs. We also confirmed these results using single-cell proteomic analysis of CTCs derived from a mouse tumor model. So we applied mass cytometry using a 29 antibody panel, which allowed us to interrogate the lineage of the tumor cells by looking for EpCAM expression, and we also looked at mTOR activity using the expression of phospho-S6 and phospho-4-EBP1. So basically, here's the results. This is single-cell proteomics of cells derived from the primary tumor and the CTCs. The pink clade basically represents the EpCAM high epithelial cells, and the blue clade basically represents the EpCAM low mesenchymal cells. And you can see that the tumor cells in these two tumor compartments is quite heterogeneous. And on top of that, the epithelial tumors basically had high levels of phospho-S6 and phospho-4-EBP1 expression in both the primary tumor compartment as well as in the CTC compartment, compared to the EpCAM low mesenchymal cells, which had low mTOR activity. Consistent with this, treatment of these tumor cells with BEZ235, a dual inhibitor of PI3 kinase and mTOR pathway, basically resulted in eliminating all the epithelial cells which had high mTOR activity and with a simultaneous accumulation of mesenchymal EpCAM low cells with reduced mTOR activity. So in terms of clinical outcome, we went back to our single-cell RNA sequencing data from metastatic breast cancer patients, and we found that elevated mTOR activity and elevated ribosomal protein expression in patients with in CTCs of patients basically was associated with poor clinical outcome as measured by overall survival and time to progression. So to summarize what I have actually told you up to now, using single-cell RNA sequencing, staining, and single-cell proteomics of CTCs, we saw that there was extensive heterogeneity and tumor cell lineage, and this was associated with the epithelial cells being more proliferative, with increased translational processes, and this was associated with poor clinical outcome. And we wanted to understand the molecular mechanism involved in this observation. So during this time, Richard E. Bright, a graduate student in the lab, did a genome-wide CRISPR activation screen to look for targets that drive metastasis. And when he analyzed the enriched targets, he found that there was increased enrichment for targets involved in protein translation. And this included known translation-related hits such as mTOR, and also structural ribosomal proteins such as RPL15, RPL35, and RPL13. These are quite interesting because they form the exit channel for the polypeptide in the large ribosomal subunit. To understand their consequence in metastasis, Richard overexpressed these proteins in CTCs and injected them into the tail vein of mice. So compared to the control cells, tumor cells or the CTCs overexpressing RPL15 and 35 basically formed more metastasis compared to the control cells, whereas the RPL13 had no effect. When he injected the RPL15 overexpressing tumor cells into the memory fat pad, basically there was no difference in tumorigenesis between the control and the RPL15 overexpressing CTCs. However, the RPL15 overexpressing tumor cells were more competent in forming lung metastasis compared to the control cells. To understand the molecular mechanism underlying this observation, we did ribosomal profiling of control CTCs and CTCs overexpressing RPL15 in collaboration with Dr. Shobha Vasudevan and Charlie, a postdoc in her lab. We wanted to look at the mRNAs that were preferentially translated in these RPL15 overexpressing CTCs. Ribosomal profiling showed us that the overexpression of this single structural ribosomal protein RPL15 basically resulted in elevated translation of multiple core structural ribosomal proteins that are involved in making the large ribosomal subunit as well as the small ribosomal subunit. So in order to see whether this elevated translation of multiple structural ribosomal proteins in the RPL overexpressing CTCs led to increased ribosomal content and global translation, we did experiments and found that in fact there was increase in the ribosomal content in RPL15 overexpressing CTCs compared to the control cells, as well as there was increased global translation in these cells compared to the control cells. So we wanted to look at the relationship of these processes to EMT. So we turned to the MCF10A cells, the memory epithelial line, which when treated with TGF-beta undergoes EMT. It serves as a great model to understand the molecular processes that are involved in EMT. So treatment of MCF10A cells with TGF-beta induced EMT, and this was associated with a decline in global translation. And when we did ribosomal profiling of the untreated and the mesenchymal cells that arise following TGF-beta treatment, we observed again that there was a strong reduction in the translation of mRNAs that code for structural ribosomal proteins, confirming all the data that I showed you previously using human CTCs or single-cell analysis of human and mouse CTCs. So we wanted to see whether increased global translation in the CTCs overexpressing RPL15 would render them exquisitely sensitive to translational inhibitors. So we did a mouse tumor model where we injected the mice with control and RPL15 overexpressing CTCs. We found that RPL15 overexpression in fact increases tumorigenesis, and we treated these mice with a combination of palbociclib as well as omacitaxin, which is a translational inhibitor. And we found while this combination had no effect in the control cells, it strongly inhibited the growth of these metastatic tumors that are formed by RPL overexpressing CTCs. So in conclusion, using single-cell analysis of CTCs enriched from metastatic breast cancer patients, we show that tumors are a mixture of epithelial and mesenchymal tumor cells. And these epithelial tumor cells are more proliferative and they harbor increased expression of ribosomal proteins, translational processes, and mTOR activity, and are more competent in forming metastases. Treatment of this mixture of tumor cells with translational inhibitors selectively eliminates the epithelial population, which is high levels of these processes, leaving behind mesenchymal tumor cells, which harbor low levels of ribosomal proteins, mTOR activity, and translational processes. However, these states are able to switch between one another quite rapidly, and as a result, these mesenchymal tumor cells give rise to a mixture again, which consists of epithelial and mesenchymal tumor cells, which then is able to seed further additional metastasis in patients. With that, I like to close and acknowledge the people who did the work. The translational work that I showed you today was done by Doug Microfluidic and Richard E. Bright in our lab. The staining work that actually showed us that EMT is a continuous process and that there's dynamic switch between these two states in patients was done by Min Yu. We collaborate very closely with Aditya Badia, a breast oncologist at MGH. And this work was done very in close collaboration with Daniel Haber and Mehmet Toner, who actually is responsible for designing and developing all the microfluidic devices that we use for our projects. With that, I like to close, and I'm happy to answer any questions that you may have. Thank you.